Hi, this is Dr. J. Bailey, and in this video, we're going to talk about writing written answers, that means essays, longs and short ones, specifically just for topic 1.4. I went ahead and took most common questions that are related to topic 1.4 from the previous past IB exam papers, and I'm placing here. We're going to learn how to respond to these questions. So here we have the first question. Most of the questions you will see, it's always, when it comes to 1.4, it's always about placing a certain type of plant or tissue or as any kind of cell in a certain type of solution. So before we get into the answers of the mark scheme for this, let's remind some things, like for example, the word hypertonic. If you remember hypertonic, it means high concentrations of solute, right? If I say hypotonic, it will be opposite, it will be low. And if I say isotonic, it will mean the same amount. That means this is usually the amount outside of the cell, this is the amount outside of the cell, and this is the amount outside the cell compared to the inside. So in this case, they're saying what will happen to a plant if it was placed into an outside situation where it has a high amount of solute. That's when it comes the next thing, where you have to know that water always move in the direction where there is high amount of solute. So it will always, water, whatever place it is, if you see a place that has a lot of salt, it will move towards the area that has a higher salt. If it's an area high of sugar, it will always move towards the area. No matter what, water always moving to a place of high solute. So by refreshing your mind on these guys, now you can see from the question, they're saying what's going to happen if a plant cell now decides to get to an area. So this is, let's say this is the plant cell, and you put in an area that has high amount of solute, because it says hypertonic. So basically you're saying, what if I put in a place where these little red dots are a bunch of salt and there's just a little bit inside? Obviously, this is what's going to happen is the water that is inside the plant right here, compared to the outside, it doesn't matter what's on the outside, but you can see that because outside there's so much solute, the water of the plant is going to start leaving the plant. And what's going to happen to the plant? The plant will start to get smaller, but we don't use the word get smaller. If you use that word get smaller or shrink, you don't get points in IB. The right word that you have to use for this in any kind of situation, it's definitely the words such as the cell will be plasmolized, or in other words, it will not have any more, the cells will shrink. So the word plasmolized is the same thing as saying that the cell will shrink. But because it's a plant, we use the word plasmolized. We do not use shrink. So just to keep here more as written here, the word plasmolized. That's how you write. Now, that's for plants. If this was an animal, then I would have used the word shrivel. But because it's plants, we have to use the plasmolized, and IB is expecting you to use that word. So this is what happens if a plant is placed into a hypertonic solution. So let's look at the mark scheme and see how you would describe this. Since also it's an outline, it probably could be up to 4 points in which you have to describe this scenario. So the first thing you have to look is hypertonic solution has more solute or higher solute concentration than the cell. So yes, the first thing you have to describe the definition of what hypertonic means. And in this case, you have to say that, oh, it means that outside there's a lot more concentration of solute compared to the inside. That's the first description. Then, because of that, you can say the water is going to move out of the cell, right? By what? By osmosis. So because water wants to go a place that has high solute, water is moving out by osmosis. Do not just say water moves out. It's good that you mention osmosis because you're getting some extra vocabulary into there. Then, since the water is moving out, what's going to happen? The water is going to go to become plasmolized. But here, they say water moves from a lower solute concentration to higher concentration. That is just basically what I was explaining the whole time, right? which it was not written because this one was saying what hypertonic is, and this is just saying water moves out. And in this one, it was actually explaining why the water is moving, because water wants to go from a place of low solute to a high. And like I said, it says reject anything related to water concentration. It's not about how much water there is. It's always about the concentration of the solute. So it's all because there's more solute out there, water move out there. It has nothing to do with the water concentration. To continue, pressure inside the cell drops, or the cell no longer is turgid, or cell becomes flaccid. So this is what you mean by the cell is starting to become less big or less this normal size. But we don't use those words. You have to use the right scientific word. So you can say, you can definitely use the word flaccid, or you can say plasmolyzed. Any of this stuff work. It's not on this one, but it's definitely going to be on the next mark scheme written. Or you can see no longer turgid. Turgid would be when it's like really big and bursts. So, or you can say the pressure inside the cell drops. Yes, why? Because the water is leaving. If the water is leaving, there's less water pressuring on the walls. And look, as I did mention clearly here, you cannot say that the cell decreases in size because it will be rejected by IB. You can also say the plant wilt. You cannot, it will be rejected. So you really have to use this scientific name, which is like flaccid. So it becomes flaccid or it's no longer turgid. You have to use a scientific name. And then you can see here, it has the volume of the cytoplasm drops, right? Or here's the most important, the cell is plasmolyzed. It just means everything starts to become smaller. But again, you have to use a scientific name. And like I said, it will be rejected if you use the word shrink or shrivel because that's animal cells. That's not plant cells. So IB is very picky with the words and make sure that you do this. So going back on this question, first define what hypertonic because it's talking about it. Then say because of that, cells are moving, water is moving out of the cells because it's going from a low concentration of solute to a high one. Then you can talk about what happens to the cell right after while it becomes flaccid and eventually becomes plasmolyzed. So you can get those 4 points in there or 5 points easily just by writing that. Now let's go another question. Here we have an image of a red blood cell. So this is we're talking about an animal cell. And here it says these images show 2 red blood cells that have been placed in a different solutions, right? It's about cells in solutions. So in this one, this cell, look at the difference. You can highlight here. You can see, look, it looks full well-rounded on this one, right? It's fully well-rounded. And this one, it looks like it has been kind of shrunk. Now, how do you know this? Because here is a normal one. This is a normal one right here. So you can compare the normal one to these two. So just by looking, we can always say, well, if he went from this to become this swelled up, it probably means the inside was hypertonic. And this one, on this case, it shrank. So that means the cell, and again, this I'm saying the inside was hypertonic. In this case, it means inside probably was hypotonic. But again, just to be clear here, okay, this is actually, I'm talking about the inside, but I can say that the solution outside here, in this case, was hypo. And the solution outside here, I can say that it was hyper. So be careful, what am I talking? Am I talking about the inside or am I talking about the outside? But in this question, it's just literally asking you to explain what osmolarity is. And osmolarity just means how much solute there is in a certain amount of solution. Okay, it's not how much osmosis is happening, it's how much solute there is in a solution. So you can see the measurement of solute concentration of a solution. So it's like how much salt there is in a liter of water. That's what osmolarity is. So make sure you do the definition of osmolarity. Now let's go for another question. So deduce, and now with reason, that means taking the gas, but you gotta explain why you take that gas. Which red blood cell has been placed in a hypertonic solution? Okay, so we can go back here, and if you remember from my reading, which one was placed in a hypertonic solution? Definitely the red blood cells 2. Why? It was because in cells 2, if outside has, okay, let me use the color here. If outside has a lot more salt, what's going to happen to the water? The water is going to leave, and if the water leaves, the cell will kind of shrink. So for sure, it's cell 2. Now, why would it be cell 2? Well, because again, it will decrease. And because you saw there that it got plasmolized, that means because it lost water, because the volume decreased. So that will be the reason. Basically, it's you're answering this question, say, why is this one? Why did you choose this one? I chose this one because I can see that the cell here kind of got plasmolized and decreased. But you got to be very careful here. It says plasmolized, which this is usually in plants, but you can also find sometimes in animal. And I would, in this case, just say that it lost water. Okay? Now let's go to another question here. We have described the process of active transport. Now, active transport, this is, let's refresh everything you need to know about active transport. Active transport, this is the cell membrane. And let's say you have molecules here, and here you have a lot more molecules inside. Okay, so there's a zillions of molecules inside compared to the outside. Now, usually you want them to go from high to low, but when it comes to active transport, you're always going from a low to a high. So even though you have a lot here, you still want to force more of these guys to go in. So when you're forcing it, you're definitely going to need something here that will push them in. And usually that will be a pump, and the pump will also need energy. So active transport is going from a low to a high, and it needs a pump and an energy. And usually we say that this is going against the gradient, the concentration gradient. That's what it usually means. So this is kind of recap of active transport. Now, it's time to describe. So you describe, what are the things you describe? The first you would describe that as a motion going from low to high, which is basically going against the concentration gradient. You can also say that in this case, it does need a pump and it does need active energy. And usually this pump here will keep changing shape to let things in. So it's changing its conformation. And when I say the word conformation, it means its shape. So let's look at the mark scheme. The first thing in the mark scheme says it requires or uses ATP, which is energy. That's the first thing the active transport do. Second, it goes against concentration gradient. This sentence you have to memorize always. Whenever you're talking about active transport, always mention going against the concentration gradient, because that is found in every single mark scheme when it comes to active transport. Okay. Next, requires a protein in the cell membrane. In this case, I wouldn't even say protein. I would say it requires a pump. Okay, do not choose channel here. And it says clearly because channel is for facilitated diffusion. Then hydrolysis of ATP. Now this is more advanced. Basically, it says to use ATP. But this, if you want to break down ATP, that is more cell respiration. If you get to that topic, then you're going to understand how you break down the ATP to get that energy. But don't worry so much about this one as long as you mention that it uses ATP. Then last involves conformation change in the pump. So the pump is always changing. That means it can be like in the beginning, it can be working like this, but then afterwards it switches and it goes down like this. So that means you're changing the shape, because the word conformation means the shape of the pump. So the pump is changing its structure. So you can see this can be a question of 4 points, and you have your 1, 2, 3, 4, 5. So you can hit them, or it can be even 3 points. So you can hit them. First, mention that it has energy, mention that it goes against the concentration gradient, mention that the pump is an example. Okay, these are some things that you can talk about. Next question. Outline, basically it's a brief question, the ways in which substance move passively across the membrane. So since we're talking about moving passively, that's when we go back and remember the whole concept of passive. Passive here, it just means that it's without energy. So passive is no energy requirements, right? No energy. Now, there's 3 types of passive, and we're just going to review very quickly so you can remember. When it comes to passive, usually we are going from an area of high concentration, and I'm going to put here as blue, high concentration. And you're going from that area to a low concentration. So you can see these three drawings I'm doing here is it going from a place that has a high amount to a low amount, okay? Now, the first case scenario, they are so small and they can pass it easily through the membrane using this guy here, the red line here, as the cell membrane, right? The cell membrane. So they just pass through. If you can just pass through the cell membrane, then you are just a simple diffusion. Now in this case, let's say these particles is a little bit big. They cannot pass through the cell membrane. So what do? You need the help. So the help will be of a channel. So you have a channel, place it here to allow these particles. And this channel will actually be specific to these particles. So this case, we call it facilitative channel. And this again does not need energy. It's just open the channel without needing the energy, right? And it's also going from high to low. This one, I put it in blue because these are water molecules. And the water molecules can easily pass through the membrane. So they're diffusing. So when they're diffusing, we don't say diffusion. We give a specific name for water. We say osmosis, which is basically diffusion of water. Now, the three of them do not need energy because they're going from a high to a low. So you have to talk about this kind of stuff. Now, another thing interesting about substance moving past is that if they have charges, right, positive and negative, they are polar, right? So that means they can easily go through the membrane. And these guys cannot, actually they're opposites, or these ones gotta be here, polar. They're positive or negative charge. They have to go through the channel because the channel will allow them to have it. These are the other ones, they're non-polar, as I was saying, my mistake, non-polar. Now these are more likely to pass through, but they can only be non-polar and small. If they're non-polar and big like these guys, they do not pass through. These ones are polar and they can be big, they have to go through. And water is small can go. So let's look at what exactly the mark scheme is asking. So starting with the first one, it says diffusion. Diffusion is a method, so you only just mentioned diffusion. You then saying that this is a type of passage transport, you already got the first point. The second, you get channel proteins for facilitated diffusion. You can say pore or channels. So what does allow? Hydrophilic particles to cross, right? Because hydrophilic, they only touch water. And since the membrane has a little bit of fat, can't go through it unless you're very tiny particles like water. So you got channels for facilitated diffusion. All of them are going from a high to low concentration, or you can say down the concentration gradient. Also, the membrane must be permeable to the substance diffusing. So imagine if you had a membrane that it's like this line and it's completely blocked. No matter how tiny the particle is, it will not go through. It just won't because the membrane is not permeable. But because it's kind of permeable, that means that means there's pores around or there's some little gaps in between. It allows tiny particles to be able to pass through. Another one is oxygen or any other substance that can pass through, like CO2, it can pass through, right? These can diffuse from even water. So mention again, as I was saying, mention an example of oxygen that is able to pass through by diffusion is important. You can say osmosis is the movement of diffusion of water through the membrane. So that's another sample. So if you saw, first I talk about the simple diffusion, then I talk about the facilitated diffusion, then I said that they go from high to low, then I talk about how substance they can diffuse, such as oxygen. I'm giving examples of diffusion, and then I talk about osmosis. Then from a region of lower to a high region of high solute concentration, higher to low water potential. So this is just talking about when it comes to water, right? What does it mean? It's not saying that going from a high amount of water to a low amount of water. In this sentence, we're talking about the solute. And we already said water wants to go to a place that has a high amount of solute. So even though there's a high amount of water, it's going down to a place that has a low amount of water. But that means, think about it. If it has a low amount of water, it means that it has a high amount of solute. And the drawing makes more sense. Here, imagine you have 1, 2, 3, 4 solutes, and here you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Okay? Now, the water is right here, and you have a lot more water here. So much water. And here you just have a little bit. So you're going from a high to a low amount of water. But looking from the perspective of solutes, here is low concentration of solutes to a high concentration of solutes. And water always wants to go to the place that has a high concentration of solutes. So are you talking in the perspective of the water, or are you talking in the perspective of, in this case, the solute? And last but not least, we have membranes are nearly always freely permeable to water, which it makes sense, right? So you basically just saying that water can pass through the cell membrane. It's the same thing as saying membranes are nearly always freely permeable to water. So this outline, it probably can be 4 points, and you can just mention that there is simple diffusion, facilitated diffusion, they're going from a high to low, and there is osmosis, which is the movement of water. Look at that, you already got the 4 points. But then you can also add examples like oxygen can pass through the membrane. And you can talk about how water is always moving to an area that has a high concentration of solute. So you can do some additional things if this becomes more points. All right, let's go for another question. Distinguish between active and passive movements of materials across the plasma membrane using name examples. So since I already explained so much the passive and the active, let's just go to the mark scheme and see what they're looking for. First one, they said passive. When you passive, what are the examples? Diffusion, osmosis, facilitated diffusion. While the active, what is it that we have? Ion pumps, exocytosis, pinocytosis, phagocytosis. Why? Because the word distinguish here, this word distinguish means what is the difference between the active and the passive. So the first difference is listing their names. Passive, let's erase here, let's put in different colors so you can see. So for passive, we're going to use the blue. Passive, we have diffusion, osmosis, and facilitated. And green, active transport, has pumps, excitosis, pinocytosis, phagocytosis. That will be your first point. Another point here is, let's look at blue. For passive methods, right, second passive method from above, where active process, second passive method above. What does that mean? It's just basically telling you, giving extra examples. Not much, this one is not much of a big deal, but it's just giving the extra examples of this or that or this or this. Doesn't matter. And then the next is passive. Passive does not require energy, while the active transport does require energy. Then you have the next one. You can see that the passive is down the concentrating gradient, while the active is against the concentrating gradient. So if I would write an essay on this one, I would focus first on telling the different types, right? What are the different types? The second thing, I would even ignore this one because it doesn't much say. Then the next thing I would do is talk about energy-wise. Passive doesn't require, while the active does require. And then I'll mention the word going down the concentrated gradient for passive and going against concentrated for active. Let me put active here. Okay, so in this case, you already have 3 points. They might ask for more. Also, for passive, we do not need any pumps, while for active, they require pumps, okay? Passive, we usually use the channels, okay? And then passive here, you gave an example, oxygen, right? While active transport, we're talking more about glucose, because glucose is a big particle, and glucose is always needed no matter what. Even if the cell already has enough glucose, the cell always wants more glucose, so you're going against the gradients. So these are just different types of things in mark scheme that you can do. But for me, first mention the different examples, then mention about the energy, and then mention about the concentration gradient, if it's going down or against, and then mention passive is more for the channels, while active is more for the pumps, and you probably already have that 5 points. I don't think they can go more than 5 on this one. So next, outline with an example the process of phagocytosis. So again, they're asking to outline and they want example. Remember to put that example. It's important for that point. And then exocytosis. Now, just as a reminder, exo means out and cytosis means a cytoplasm. So in this case, what they're trying to say is something is leaving the cell out, right? So that means we're probably going to have a vesicle inside of the cell that is moving in a direction to release the stuff out. That's what exocytosis is. And we're going to see examples depending on what could possibly come out from example from whatever your studies are. So let's look at the mark scheme. The first thing is the students don't even realize is that you can say vesicles carry material to the plasma membrane. That is so obvious. Now, could you, some students were asking, Miss, could I actually draw it? Yes, you can. You can draw it and put lines. They will accept that too. So first, the vesicles are carrying material to the plasma membrane because, again, it's going to leave the cell. So you have to mention that it's going to the plasma. Vesicles are moving towards the plasma membrane. Next, once it gets to the plasma membrane, you have to mention that it's fusing because the vesicle will join, or should I say, the cell membrane of the vesicle will join the cell membrane of the membrane. Then, which basically is the next one. See, it joins the phospholipid layers. So remember, the phospholipid bilayers of the vesicle will combine with the phospholipid bilayers of the membrane every time things are popping out. And that can only happen, why? Because the membrane is fluidity. It has fluidity. If the membrane was not fluid, you couldn't do that, okay? Next, and once that happens, the material is released or expelled out of the cell. So it's the same thing if I had the vesicle here, right? And let's put this is the cell. So you can visually see what I'm talking about. See, all of this is just a description of what I'm going to draw here. So this guy is, vesicle is moving in the direction of the membrane. And then once it gets there, this vesicle, let's erase now. So it's moving, right? It's moving, and then it reaches the corner here. Once it grown here, it's going to fuse with it, which that means what? Its cell membrane right here, right, is going to fuse with the cell membrane of the cell. And this can only happen because there is a fluidity. That means you're allowed to open close. And whatever particles that was inside here will move out. Now, when they join together, this is what the cell is going to look like. This is the cell, and look, the membrane of the vesicle now became part of the membrane of the cell. That's what it means by fusing it, by joining the phospholipid layers. What else do they have? The membrane flattens, which makes sense. Look, the membrane flattens, right? Because before it was a circle and now it flattens. And then you got to give an example because here is asking. You can talk about the exocytosis of a neurotransmitter. You can talk about hormone secretion, endocrine secretion. There's so many. And right now you can't think of, but once you get to the human body, which is topic 6, you kind of already start having more ideas. Or you can even talk about glycoprotein, which is coming out of the Golgi to go outside and become an antenna. So there's many ways you can give as an example for these. So moving on, let's go to the next one. Oops, this is carry on. Describe the transport across the cell membrane. Now, when I talk about describe a transport across the cell membrane by osmosis, we want to explain what osmosis is. And I already explained many times in the previous questions. So let's go straight to the mark scheme. The first, it's a form of diffusion. So you're defining what osmosis is. Most likely the next one is going to talk about the water. See, yes, osmosis is by the movement of water. Next, you're probably going to talk about how the membrane is partially permeable or selectively permeable. That means it allows water to pass through because it's kind of permeable to water. It's selective. It allows water to go through. Then you got to mention that it's going from an area that has low solute to an area that has high solute. Or you mention in the water perspective, the water is going from a high concentration to an area of low concentration of water. So be careful. Are you describing the solute or are you describing the water? Because if it's water, it's high to low. If it's solute, it's low to high. So you got to be very careful in describing this. So you can see that it's very easy. And there's one more thing. What is osmosis? It's passive. Aha. And it's a passive. It does not need energy. So this is a very easy one. First, describe what it is. Mention about the water movement. Mention that the membrane allows it because it's semi-permeable. Mention either way. Talking about the solutes or the water. But make sure you're putting the right letter if it's high or low, depending if it's salt or the water. And then you mention that it's a passive with no energy. You get the full points. And one last thing that I forgot about osmosis is that this is if you're doing HL kidneys, you can always talk about aquaporin channel proteins. But if you're SL, don't worry so much about this. This is more like an HL answer. And that's pretty much for 1.4. Now here I have some extra questions that I have not seen pop out in IB exams in the last decade, but could be coming. Make sure you know how to define pinocytosis and phagocytosis. Explain how salinity of an organ is important during surgery, because as much as you have to know why we do it in surgeries, I doubt it. I hardly saw questions about it. List two substances that can go through facilitated diffusion. The facilitated diffusion only allows large particles. Examples could be, I don't know, in this case, it can be a specific hormone that is a protein hormone. So make sure you know two of them. And last but not least, talk about the endomembrane system function where the, secreting proteins. That means talk about these guys, how they work with vesicles, how the rough and the smoothie are. They make the carbs and the lipids and proteins, and then they combine in the Golgi, and the Golgi, it sends a vesicle out to the cell membrane. Just make sure that you go through my lecture videos, because I'll be able to answer some of these questions. So be prepared. IB can always pop up with new questions. And that's it. Thank you.